So I want to talk about a story that's related to understanding the basis of adaptation. Like Christian, let me just get a drink of water here quick. Like Christian, I'm basically trained as a population geneticist, and it's been a really exciting time to try to think about using variation in natural populations to make inferences of how adaptation has occurred. I mean, originally mostly focusing on individual samples from different populations, now much more focusing on, for example, what Chris has been and you all are doing, experimental population and following that. But I want to talk about particularly untangling the influence of genetics. It turns out also ecology and function in shaping the very rapid, surprisingly rapid evolution of a particular set of genes that are absolutely essential because these are the genes that control the production of gametes. No gametes, no reproduction, end of species. Let me see if this works. Okay, so, but this raises, of course, the basic question, which you guys have thought about too, is how should we study the genetic basis of adaptation? Should it be top down, start with an interesting phenotype and go down, or and looking at the phenotype seen by selection to try to figure out what the gene is, or should we go the other way, bottom up, what I call, where we start with a signature of selection in the gene, a prediction of a target of selective sweep or a particular McDonald-Kreitman test, a very strong protein differentiation. And can we work backwards to figure out what is the phenotype that's actually seen by selection? What is the natural selection action that is, in fact, influencing this dramatic change we're seeing? Now, there's a lot of good examples of these two approaches. Certainly, the top-down approach of the phenotype being seen by selection, it's hard to particularly striking examples, visually striking in terms of things are the finch, or Darwin's finch's beak, and also pigmentation in, for example, and this is just, is this working? There we go. In beach mice, also in mice in Nebraska, where you have strong correlates between the habitat, environment, and the coloration of the mouse. If you mismatch your coloration, you get eaten by hawks or whatever. And these both situations, people have been able to take and narrow it from the phenotype all the way down to the underlying genotype. The genetic variants is specifically responsible and show the selection coefficients associated with those variants. So that's been great. The other approach, and so there are some systems we could study that too, and I guess I found out yesterday that it's Krampus today, so happy Krampus Day if that's what you do here. I don't know. Anyway, this was in Salzburg a couple of days ago. We ran into the Krampus walking through the Christmas markets. It was pretty scary and dramatic. These guys are weird. Anyway, so this would be a great phenotype to study, but we only got DNA samples 2 days ago. So we're going to focus on another kind of approach where we look at the signature of selection of the gene and try to look up at the phenotype. And as I say, this is something that is quite complicated. You guys know that too, and I will just illustrate that. But to me, about 13 years ago, I decided we've been making so many predictions of where selection was acting in the genome. We were starting to increasingly realize the complexities of disentangling demography and so forth from natural selection. But I got worried about it. But I also felt like, look, we have to stop just making predictions. We need to test those and see if we can find what is functionally different among these populations or species in terms of what's generating these targets of selection. Now, again, what's happening here? Put new batteries in this, so it should work. There we go. So, can we do this? How do we do it? And what is natural selection actually seeing? And so, our focus is not on Krampus, but on Drosophila, because in fact we have this enormous amount of differentiation. In fact, if you consider the number of generations per year and so forth, there's more within the genus Drosophila, there's more evolutionary, kind of replications and, generations than all placental mammals. So if you think about it, that's a dramatic amount of evolution. And yes, these are all diptera flies. They look quite similar at our level, but there's really a remarkable phenotypic and genotypic diversity and ecological diversity amongst these different species. Now, of course, there's about 1,600. These are just the 12 original genome sequenced. So there's a tremendous number of different species that allow us to actually go in and look at population samples and make predictions of where natural selection has acted and when, and so forth. And so this has been the system that we've focused on. Now, we had focused on, for a number of our studies, genes specifically involved in reproduction, because that's, of course, where the rubber meets the road in many respects. If things influence whether one male mates successfully with a female versus another, or in fact, whether the eggs and sperm actually specifically unite. I'm going to switch. You had a point or two here, Joy. We and others had shown that there's a lot of positive selection acting on either the male reproductive genes being passed to the female. Here are the so-called seminal fluid proteins, also the proteins that mediate the interaction between the mammalian sperm and the egg. Lots of very strong positive selection going on there. And there's been lots of debate about what actually causes this. Is it associated with sperm competition, sexual selection, male-female contact, conflict, and so forth? I've always been intrigued by the possibility that it's really the interaction with microbes and immunity that also play a role in driving the receptor sequence evolution on the female side that causes the male to have to catch up to say. So there's lots of different opportunities there. And in fact, there's an enormous number of really important and striking studies of that make predictions that particularly fit with a male-female conflict, but also sperm competition. But it's often very hard to actually follow up and validate those. Just because it fits the model doesn't mean that necessarily explains things. But I think many of the studies are really extremely solid and important. Kristen's group, you've contributed to this as well. But what about if we take and look at proteins that are involved in genes that are involved in getting to that point of males and females actually reproducing? And we thought originally that going kind of upstream, as I call it, in the production of gametes, we would get to be many more conserved genes. Much to our surprise, though, when we focused on these genes, again, the germline stem cell genes, the immortal cells, so to speak, that are responsible for either renewing as germline stem cells or, in fact, differentiating to form eggs and sperm, that they're all premeiotic. They're genetically identical. There's no interaction between males and females and no competition between different males. So we kind of thought, okay, there shouldn't be any really much positive selection going on at all. We might see one or a few genes which would simplify any of our analyses. So all these traditional evolutionary conflicts seem to be absent. Well, let's look for a minute at what these genes are. So again, the germline stem cells can be either found in females, in the ovaries, in the tip of the germarium here. Essentially, these are GSCs, or germline stem cells here, and also in the tip of the testes here. And these can either self-renew and stay as a germline stem cell, or they can, in fact, get an asymmetric division and move away from the cap cells up here, and actually we'll see signaling changes, and this becomes a cystoblast that then undergoes a series of mitotic divisions and then meiosis to form an egg. On the sperm, on this side, basically we get these gonioblasts, which then go into spermatogonia and form sperm. So this is the origin of all these gametes. And what's striking is that we actually know quite a bit about the number of genes and the particular genes that are involved in this. There's particularly the best knowledge of oogenesis really of any species is known from flies in females, because First off, unlike mammals, oogenesis continues through the life of the fly. You continue to produce eggs and so forth. And we know at least some of the major players here. These are some of the genes that are involved in regulating a lot of this process. YB is an essential gene for connections to the germline stem cell, to the cap cells here. Genes like PWE are involved in piRNA selection transposable elements. A number of these other genes, in particular BGCN, nanos, and so forth. Nanos is a self-renewal factor. If we, in fact, move and have an asymmetric division, we move farther away and we have some signaling that changes. And now this gene called BAM, which we'll see is what we'll find out what it means in a minute, partners with BGCN now to suppress a couple of other self-renewal proteins, and we end up getting differentiation. So it turns out this BAM is the absolute critical switch gene for oogenesis in flies. Now, this is a protein coding gene, 442 amino acids long. It doesn't have any, classic structural domains. What it does have is what's called a PEST domain down here, a high frequency of the amino acids PEST, and this is typical of proteins that get degraded very, very quickly. There's studies have shown that there are a couple of regions that bind other interacting proteins, CAF40 binding here, the BGCN partner binds here, CNS4, another partner protein binds across the same region, and it's been shown that ubiquitin binds, or this region here binds ubiquitin as well, which we'll return to later. So this is a 442 amino acid protein, reasonably sized. The thing is that this protein here, as we'll see how it folds up in a minute, it seems to act like a light switch, and it basically brings together other proteins, and it acts like a light switch, like a light on a light switch in a room. This is wired in such a way so that one direction is on, and if it's on, we get differentiation. And the only way the gene comes on is if you move it away from signaling that comes from these cells here. And I'm not going to get into the details of that. It's not useful for you guys to understand that. But it is a critical thing is that when it's on, that actually then promotes differentiation. So it's without it, we end up with what's called a bag of marbles. And as you know, you guys all work in Drosophila, so you understand that Drosophila geneticists like names that mean something. So this is basically looks like a bag of marbles. So that's because it's basically, these are essentially germline stem cells that just keep dividing and forming more germline stem cells. So there's no differentiation at all because the bag of marbles protein is deleted in this particular mutation. So I'm going to refer to this gene a lot of times as BAM, B-A-M, because that's the abbreviation for bag of marbles. Okay, so what was striking is that we took a look at this protein because we were struck immediately when we first did some sequencing that of samples of populations that there were a lot of differences between melanogaster and simulans between these two proteins. If we looked at actually the number of fixed amino acid changes across the whole gene, on average, we found as many as 60 amino acids or 59 amino acids fixed between melanogaster and simulans. So that's out of 442 amino acids, that's a huge number of amino acid changes. And if you do a McDonald-Kreitman test, which is basically looks at the number of polymorphic synonymous versus non-synonymous, if you use this as kind of a metric of the constraint, non-synonymous, there's fewer polymorphisms here than there are synonymous, and these are raw numbers, so we'd actually expect to see 3 times as many here as here, but we see fewer. But we look between species, we see many more fixed amino acid or non-synonymous changes than synonymous, which is indicative of an acceleration of amino acid differentiation or replacements in that lineage. But this pattern of positive selection is found in some species. Melanogaster, several populations of melanogaster, only some populations of simulans, not others. We find it in Yacuba and Santa Mea, but we don't find it in Tessieri. We don't find it in Ananasti, at least this population of Ananasti. We do it in a different population. Pseudoobscura not, Moavensis no. So there's this kind of heterogeneous pattern of positive selection. So it's not just being a stem cell that makes this be rapidly evolving. There's something about interaction in individual species with some conditions that lead to positive selection, some of those, but not in others. Again, this is selection for diversification of the protein sequence in all these cases. Now, we decided to take and focus on this gene because at the time we started, it was basically thought to only function in germline stem cells and be expressed in a very narrow window of only basically 4 cell stages, very few cells at 4 cell stages. And we thought, okay, this will simplify understanding the function. You've got a candidate, very rapidly evolving gene. It's targeting the amino acid changes, so we know it's in the coding sequence changes or what's under positive selection. And the hope was by picking this gene found expressed and only functioning in this one tissue, we reduce the amount of pleiotropy and epistasis with other kind of phenotypes. Okay, that was great 10, 12 years ago. Well, 7 years ago it was revealed that it also plays a role in hemopoiesis. These are the production in larvae of blood cells. But here it's interesting. The light switch actually for hemopoiesis got wired upside down. So if you turn the BAM gene on, it actually stops differentiation in hemopoiesis. So interestingly, if you in fact have a deletion of the bag of marbles gene in, for example, in melanogaster, you basically just all the stem cells basically differentiate, which is fine because you end up with lots of blood in the larvae, which then undergoes, metamorphosis into adults. And even more recently, it's revealed that in the gut, basically BAM, in fact, when it forms a ubiquitination complex with OTU in the gut to basically regulate immune homeostasis, and that can influence lifespan. So over the time it's taken to work on this, we've gotten some complexities to this, but actually it turns out that this role in ubiquitination may actually be related. And what ubiquitination is basically if you take proteins that are targeted for degradation, you attach ubiquitin proteins to them. In fact, that targets them to go to the proteasome to be degraded to basically be recycled. So if a protein is targeted for recycling, it gets ubiquitins attached to it. But you can also have some proteins that go in and take ubiquitins off that other complexes have put on. So you can gives rise to a balance, and that actually is the role that it plays in the gut in terms of maintaining homeostasis there. And actually with functional BAM, it actually leads to extension of the lifespan. We're in the middle of, well, we'll talk about that later. So anyway, it raises these questions though of what actually is causing this burst of rapid selection at BAM. And one of the questions is, has BAM function been changing between species? Again, we've often been in the mindset that, okay, we study function in one species, the key model species, and it's likely to function in the same way in all these other species. Well, that's actually might not be true. And maybe there are functions in certain lineages that need evolution fine tuning. And so, in fact, there's strong selection going on there. And that may be what's giving rise to our patterns of bursts of positive selection. It could be changes in function due to selection for changing reproductive schedule. This is the ecology part. So, of course, reproduction, as you guys know, I mean, you can produce all of your progeny early in your life, some spread out over the whole course of life, or some only towards the latter part of the lifespan and wild populations. And BAM could play a role in the timing of expression. And you can imagine that in some sense, in stable environments, Getting all your progeny out all at once could be a good thing, but in an unstable environment, dumping all your progeny out at once, if you happen to dump them out the wrong time, that's not a good thing. So you can see selection could act there. So automatic, or kind of very quickly, you're starting to see our simple worldview of maybe we can track these down in a very simple way to a single phenotype is going to be more complex than we hoped. Anyway, and it could be that, there are constraints, sorry, it should be a T there, of the germline switch role means stronger selection for other functions, which could act on BAM in the same way. Okay, so the first thing we did in finding this result was to say, what are the consequences of all these amino acid changes? Does it have any functional consequence, and is it males or females it affects? And so at this point, almost 8 years ago or so, what we did is take and generate a transgenic fly using site-specific integration. We took the melanogaster BAM out of the second chromosome or the third chromosome and put it into the second chromosome, and then we could cross a null into the endogenous third chromosome site and study the effect of the Drosophila melanogaster BAM. And as a control, we put that back into flies and looked to see whether fertility was normal, and also the number of mitotic divisions here. You can see these are increasing from the germline stem cell, BAM labeled in blue here, and in fact, the number of different cysts basically here increasing. These are undergoing mitotic division of the nuclei, but not of the cell. It stays still in one cell. We end up with a 16-cell basically cystoblast that then undergoes meiosis to form essentially a single egg. If we take simulans though and put it in, the result was that we had reduced fertility, somewhat reduced. We still had fertility, but reduced, and we see now asymmetries in the mitotic division. So it suggested that there really is a consequence of all these amino acid changes, but remarkably, it didn't ablate function. So it still seemed to be able to function to some degree. The important thing was that it basically led us to focus on the female side of things rather than the male side, because we saw no difference in terms of the male, either the cytology of spermatogenesis or in terms of male fertility. Excuse me. Now, you might be asking, well, with 60 amino acids, does this change the structure of the protein? And only recently we've been able to actually explore this, because the protein has not been able to be crystallized and the structure determined. But it turns out with AlphaFold, we were able to make predictions. And this is the AlphaFold prediction for melanogaster matched to Drosophila simulans. And so you can see that a lot of the molecule actually matches up reasonably well in terms of these. There's a series of helices here that actually are quite virtually identical if you line them up. The disorder regions actually out here, these are just guesses of what you don't have a prediction for those. But the key thing is there are a couple of core regions that in fact are predicted to actually remain structurally quite similar. So it's consistent with the idea that it still functions pretty well, but not necessarily exactly the same. I'll return to the fact that we do have predictions of some of these core helices here, because the predictions for these helices here are actually very strong predictions. So, and we've kind of confirmed that by taking and looking down different lineages of melanogaster, simulans, others, and looking for compensatory changes that would maintain the contacts between these helices, and we see that, evidence of that. So it gives us confidence that this is not just some wild pipe dream of what the BAM protein looks like structurally. Okay, we did decide, because in site-specific integration using at P sites, for example, we're taking the whole gene, including synonymous sites, introns and so forth, even though they're small, and putting them into melanogaster. We have shifted now to using CRISPR-Cas9 to edit basically every single change we want and compare those. So from now on, all of our studies have used simply CRISPR-Cas9 to make every single edit that we want in Drosophila melanogaster. We're using edits into stimuline sequences or other sequences in other species. The challenge with this has been that, okay, you can get CRISPR to work in other species. It turns out to be a little tricky for some species, but we've been able to figure out how to get to work. The problem is we don't have things like chromosome balancers. And this is a gene that if you make a null, essentially it's sterile. So if you make a null for this in a, in simulans, for example, or ananassi, it's going to be sterile, and a sterile fly doesn't reproduce. So you don't have the stock. You can't maintain a stock. So the trick we came up with, and this was led by Jackie Bubnell, who's now a postdoc in the group, was to take and insert disruption, a 3xp YFP, or as we'll see another fluorescent protein in the first intron, and also or exon rather, that also introduced a stop codon. And in this way, we then had a stock that we could maintain as a heterozygous stock, and that was fluorescent, these yellow fluorescent eyes. Okay, that's one, but what else did you do? Okay, well, the trick was to make a 3xp DS red insertion as well in another set. So we maintain these as two separate lines, a yellow line and a red line. We have to constantly select these periodically and maintain in there, because otherwise the wild type chromosome will take over. But if we want to study the phenotype, we just simply cross these and we get kind of orange-eyed flies here that in fact contain both of these alleles, and we can study that. Of course, these are sterile, but we can study the phenotype. We can study what effects the alterations have on on the phenotypes and other things. So this has actually been a very valuable tool. We've done this now for a variety of different species. And I want to take you through in a quick tour the results that have emerged, because they're really kind of striking. So first we did the control, essentially. We looked at melanogaster here and Jackie Bubnell here. This is an image I was down in the imaging center with her when we looked at the first null lines, and it actually came up, it generated a null, which is what we were hoping for, because in fact, if you take and look at the wild type, the YFP over wild type, the DS red over wild type here, so these have at least one wild type chromosome, BAM being, this now has the YFP over the DS red, so in fact there's the two disruptions here, and in fact the fertility is 0, so these are sterile flies. You can see the difference relation. It's highly significant here between these. And so this was, kind of confirmation that yes, if you make nulls of this homozygous, no progeny being produced at all. We looked now at also additional species we looked at simulans and Yucuba, and again, and importantly, we see it not only in terms of fertility, but we also see it in terms of cytological phenotype, that if you, in fact, look at the nulls here for these, the nulls basically, unlike being the wild type or heterozygous here and seeing lots of differentiating cells here, we actually basically have only tumorous ovaries here in the null. And if we look at fertility, we also see that fertility is also, because I don't have that slide, the fertility here for these is also essentially there's no progeny being produced by simulans or Yucuba either. Interestingly, if we look at Drosophila tessieri, this was a total surprise because if you delete the gene in Drosophila tessieri, again, this is a relationship here to Yucuba, very close to this group here, and Yucuba, we in fact see we can delete that and look at both females and in males, and we have, excuse me, normal progeny. So here's an absolutely critical gene, yet we delete it so it can't be produced, and this key switch gene is now gone, but yet we still are producing progeny. So wait a second, what's going on here? Further interestingly, if we take a look at Ananassi, this is a further twist, a little more further out in the Melanogaster species group here. But in fact, we do see the expected phenotype of sterility in females, but males now are fine. So wait a second, what's going on? We've got these complexities of this regulation that's going on. This was published in MBE last year. And so this kind of led to this kind of striking pattern that, in fact, for melanogaster simulans and Yakuba, if you knock out this gene, you actually get tumorous ovaries um, uh, and testes, and you get no progeny, which is like melanogaster. Tessieri, though, you can knock this key switch gene out, and the lights are still on. Okay, Ananassi, the lights go off in ovaries, so to speak, but they're still fine in testes. So all of a sudden, there's this pattern of, again, kind of related to what you guys think about as kind of genetic redundancy. You got the same; these are still producing, Tessieri are producing gametes, Ananassi is producing gametes in both males and females in the wild, but this key regulation of it seems to have changed. So of course, there's lots more species that one can look at. The number of species is growing dramatically. These are ones for which we've got really high quality genomes now. So we're in the process now, Luke Arndt's particularly with Jackie, generating nulls and additional other species to actually try to see exactly what the pattern, the sequence of, where has BAM's role been acquired? Is it new? Certainly we know it seems to be new in the genus, but how much heterogeneity is there in this regulation across the genus? And again, we're also taking a look at the presence of germline stem cell genes. I'll return to in a little bit across all these species, again, to see how much flexibility in not just bag of marbles, but also other genes as we go across. And as a prelude, we know that bag of marbles gene is present in all of these species, but we only know in some species that it's not actually critical to this particular process. We're testing it now in the process to test for the influence in terms of stem cell function in hemopoiesis and also in gut homeostasis, for those we don't know yet for that. Okay, so again, this question is, is it a changing function? We'll learn more about that as we get more nulls to evaluate, to be able to get enough information to make a power prediction about, is it changing function? But right now, it doesn't seem to be, the changes in function don't seem to match the species that, in fact, are under positive selection. So it's not looking like, in a simple sense, BAM's function change is, in fact, what's driving the pattern of protein sequence evolution that we're, in fact, have been observed so far. So it raises another question, could it be due to some sort of evolutionary conflict? There really is an evolutionary conflict going on. And there are a number of sources that could be causing conflicts in the germline. So it turns out that there are a variety of things that are trying to get into the egg, essentially, to get passed on to the next generation. Transposable elements, viruses, endosymbiotic bacteria. There's a bacteria called Wolbachia we'll talk a lot about. This is a alpha proteobacteria that only survives in insect cells, essentially, and is kind of an interesting one we'll talk about. The question is, could gut microbiome be playing a role? Could it be sexual conflict in terms of timing for germline stem cell differentiation? These are all open questions. We are going to focus, though, on the endosymbiotic bacteria Wolbachia for a particular reason. And that is that this intracellular bacteria is, in fact, a dramatic reproduction manipulator in insects. It's found in, on average, between 60 to 70% of all arthropods. It's, in fact, inhabits the germline. It's also found in some other tissues as well. It's purely intracellular, maternally inherited, and it turns out to be a key manipulator of reproduction in females. If you take a female that's infected, mate it by a male that's uninfected, essentially all the progeny will be infected with Wolbachia. It will transmit then, and it increases frequency through populations. Because of this trait, actually, it's been the melanogaster Wolbachia has been introduced into mosquitoes, for example. It does the same thing as it does in many Drosophila species. It manipulates reproduction towards the production of females that are all infected, spreads the infected females to the populations, both males and females. You might think, well, why would you want to do that? Well, it turns out Wolbachia, one of its other traits is that it, in fact, inhibits, by a variety of interesting people are just working the pathways out. Now, it basically inhibits the reproduction of viruses in these in Drosophila, but also in mosquitoes. And the intriguing thing there is in mosquitoes, you might think, well, why would inhibiting viruses be useful there? Well, actually, dengue virus, chikungunya, a couple of Zika virus, those are all viruses that, in fact, if it's a Wolbachia infected mosquito, they basically won't, those viruses don't survive in those mosquitoes, and then they won't transmit it. So this is a very, in fact, important and powerful potential biological tool. It's being implemented right now as a way to control the viruses in populations of mosquitoes around the world. Our interest in this is, okay, well, that's great. We don't really know still how it manipulates interactions with the male, with the fly genome. And so we'll return to that in a minute. But this is why there's a lot of interest biologically in Wolbachia. Now, this is where it gets interesting. That we actually know that Wolbachia directly interacts with bag of marbles. And how do we know that? So this is a study, a thing that Heather Flores did when she was a graduate student. We actually there's a BAM mutant that was been in the literature for a long time. It's a single amino acid change in bag of marbles, but that amino acid change, even though it's just a single amino acid change, actually means, excuse me, that the function drops quite dramatically compared to what is normal. So instead of normally being produced for this particular genetic background, about 120 or so progeny, it only produces about 45 to 50 progeny. So it's a significant knockdown, evolutionarily certainly significant. What's intriguing is if you take that line and infect it with Wolbachia, now the fertility increases. So you get the fly sick and it gets better. So it's kind of a bizarre interaction here with presence of Wolbachia in that mutant stock actually rescues not all, but a lot of the fertility in that thing. So clearly, there's not a general interaction between Wolbachia and BAM. There's quite a specific interaction, specifically, and also one that affects the function of the bag of marbles gene itself. Okay, so, and again, another reason why Wolbachia is interesting as a candidate for the positive selection is that we see Wolbachia is actually present quite heterogeneously across the genus. Many species don't have Wolbachia infections. Some have actually quite common infections. So there's not a good correlation at this point between the presence now of Wolbachia. The challenge is this is like studying the effect of, what's the pattern of sequence or immunological genotypes of people that are associated with having the flu right now versus, the thing is, if you just measure the people that have the flu right now, that doesn't count the people that had the flu last year, the year before, the year before that. So we've tried to use the kind of genomic integrations of Wolbachia into the genome as a way to kind of look for past infections, but it turns out it decays too quickly. Wolbachia does, it turns out, little pieces, in some cases, integrate into the host genome. And we were hoping to use those as kind of a genetic fossil, essentially going back through time, but it's not turned out to work quite so well. Anyway, so what we have asked about is, what if we look at different Wolbachia? Is it really a very specific interaction? And it turns out that it is. We needed to have a homogeneous background, not the original mutation. So Jackie actually made a mutation that in W. white 11 melanogaster that has the exact same amino acid here, but the rest of the genome is completely homozygous. And this allowed us to actually test for differences of different Wolbachia too, in terms of how they influence things. And we were able to, I'm going to skip this here, but actually find that of the different kinds of Wolbachia, things that tend to have a low level of Wolbachia actually had, in fact, different levels of rescue of the phenotype. The ones that at the highest presence of Wolbachia in these Wolbachia phylogeny here tended to actually have the strongest rescue effects. And importantly, Wolbachia did not have any effect on fertility of wild-type flies. So it's clearly an interaction between Wolbachia and Bagomorphals in a very genotype-specific way. We've also taken, to simply give a quick perspective on other approaches we've taken to ask, is it specific to particular regions of Bagomorphals? The original mutation was here. Mabel Wenzel generated a whole series of using alanine scan, inserting alanines, strings of alanines across the protein here, and asking, did these produce mutants? And if they did, are they rescuable by Wolbachia? And interestingly, it kind of clusters, particularly around this ubiquitination domain, many of the mutations that are associated with having a phenotype, fertility phenotype knocked down in these mutants, but that is rescued by Wolbachia. And this, again, is another thing that will point to a role of regulation of ubiquitins potentially in this whole process. If we look at where those structural proteins, the changes are found, many of them are found directly in this region that is associated with binding to ubiquitin and in these cluster of helices here. So this is not, the structural approach has been less effective, but it's been kind of reassuring that we're getting changes in parts of the protein that make sense in terms of what we do know about these molecules. Okay. So again, with these different regions here, there are lots of different, I've given you a very simple view of what's going on. There are actually clusters of genes that all interact together here that associate with binding here, clusters of genes that are involved in each of these other complexes as well. So there's a big, rich complexity to the underlying biochemistry and cell biology going on here that I don't want to get into. I want to focus on the overall general point that, in fact, different variants of Wolbachia rescue the hypomorph to different degrees and rescue different hypomorphs to different degrees. And it does raise the question about, okay, well, is there a change? Does Wolbachia, how does it cause these changes? Is it changing the expression of genes? There's lots of levels of control. One thing is to ask whether it's changing the level of expression of genes in the hypomorph, and can those proteins that change, or the genes that change, can those help us understand what the actual mechanism, in fact, of this manipulation of reproduction actually is? So very quickly, you guys, many of you are familiar with RNA-seq studies. So we actually have taken and looked at flies unmated with 3-day-old, mated 3 days old, mated 6 days old, mated 9 days old, did all the controls and so forth, lots of replicates to actually take and look at what was the best time period. We realized from these studies that, in fact, using, again, the typical pathway of analyses of these, the different, groupings here and biological replicates here, essentially looking, and mainly I'm going to focus on comparisons of Wolbachia infected to uninfected Drosophila here in terms of differential expression of genes, and to basically hopefully highlight the genes that are solely impacted by Wolbachia infection. We also factored out genes because this is a Wolbachia infected and uninfected, and there's a difference in tissue size and so forth, so we had to factor those things out. This is all work with Catherine Kegman, who's just finishing up and defending actually next Friday her work on this. And from this, to identify candidate genes that are actually involved in this interaction between Wolbachia and Bagomorphals. And it turns out that, interestingly, of the genes we looked at, bag of marbles is not affected in its expression at all, which is was a bit of a surprise to us. There are a number of other genes that are affected by the uh, presence of Wolbachia in the hypomorphs. So, and what's actually being rescued, interestingly, BAM's partner BGCN is in fact the most highly upregulated gene in the mated 3-day-old flies, which actually we think are the ones that are the most priming things for rescue of this fertility defect. We also see a variety of other genes that some go up, some go down in expression, but when we take and focus on the genes that are involved in this, process of either self-renewal or differentiation, again, BGCN is one of the key players in this that is really partners with BAM. So this was an intriguing result. Understanding what that means is still a little bit unclear. What we have done, though, is take us and focus on the groups of genes that are particularly altered in regulation, both up and down together, and ask what kind of processes are highlighted. And this is work that we're just really in the middle of right now. So this is all kind of very preliminary kind of thing. But perhaps the strongest process that seems to be influenced by these changes in gene regulation are genes that are involved in ubiquitination. And so this is, again, involved in genes that are associated with the turnover of other proteins and regulating and either deubiquitinating, I mean taking the ubiquitins off and keeping the proteins expressed longer, or actually having some proteins sort of be degraded faster than we actually might have thought. And again, these are all candidates for targets for what Wolbachia is doing to manipulate the expression of Drosophila genes and causing this specific BAM-specific interaction that we see with regard to the rescue of the mutant phenotype. And again, there's a couple of different ways in which BAM plays a role, one of which is to interact with a protein called OTU that basically it takes cyclin A and takes ubiquitins off cyclin A. So it now actually leads now to germline stem cell loss or increased differentiation. So this is involved in the maintenance of germline stem cells, which is obviously critical because we have to maintain germline stem cells. If all of our germline stem cells differentiate, then we're going to run out of the ability to produce new progeny, new gametes. And so, but there's also a role that BAM plays itself, is that BAM itself is ubiquitinated. And so, in fact, when it gets ubiquitinated, it gets degraded. And this actually is associated with germline stem cell differentiation, or basically blocks germline stem cell differentiation when you degrade it, because it basically keeps it. So these two processes that both involve attachment or removal of ubiquitins seem to be, in fact, where Wolbachia is manipulating reproduction in insects associated with at least the bag of marbles gene, and potentially in other situations. So we also see things that are involved in histone lysine methylation and autophagy, basically the kind of we're basically parts of the cell basically and individual proteins will actually sort of degrade and kind of get renewed essentially over time. So we're making progress towards the functional understanding of what's really the interaction between Wolbachia and bag of marbles. And we don't know, though, is this actually driving the pattern of positive selection at this point. So Wolbachia does not alter transcription of BAM. It does alter a number of other genes. And so at least we're starting to get a hint at what's actually going on. But again, underscores the complexity. Even in a gene as simple as bag of marbles, understanding exactly the nature of positive selection on it and what's driving that is still kind of eluding us. This did lead me back to population genetics, though, because one thought that we had was because Drosophila species and insects frequently, even though they're infected with Wolbachia, they lose Wolbachia. Maternal transmission is not 100%. So one thought we had was that, okay, if you're infected with Wolbachia, now it buffers you against mutations like that hypomorph. So maybe in fact, when you have a Wolbachia infection, you can accumulate harmful deleterious mutations, amino acid changes in the protein, and you still reproduce fine. But when you lose Wolbachia, now all of a sudden there's a scramble to get back to a functional protein. So we posed a simple model working when Miwa and my lab, together with Runji Shen and Philip's lab, we worked together, approached them about collaborating, and they did, to set up first an arms race model, a classic arms race model, to kind of get the predictions of what we'd expect there in terms of there was really some sort of cat and mouse game or some sort of arms race, because BAM's starting to evolve away from the influence of Wolbachia because yes, it manipulated reproduction to generate more fertility, but maybe at the wrong time or things like that. The question was, if we're having positive selection here, either associated with change in function or some sort of conflict between Wolbachia and bag of marbles, we wanted to get predictions for the patterns of selection over time there. We also then looked at, so here you're under positive selection when you have Wolbachia to escape Wolbachia's effect. When you lose it, there's no positive selection at all. The second model was this buffering model, which is kind of a simple model. Basically, that if we have Wolbachia, it kind of buffers it so we can accumulate deleterious mutations over time. But then when we lose Wolbachia, then all of a sudden there's strong positive selection to return to the optimal phenotype. So we wondered whether this process actually could, in fact, cause the positive selection we're seeing. And we didn't have to invoke an arms race, but maybe it was just this buffering of Wolbachia back and forth. Basically, the fundamental question was we could, in fact, and I'm not going to go through it's a very complex model in terms of the selection coefficients and everything else. But it turns out both models do predict positive selection. So actually this buffering model can predict positive selection on a protein. There really isn't any positive selection for a new function. It's just simply periodically recovering from the accumulation of deleterious mutations by drift. But in fact, what we found is that the amount of positive selection that we saw and the particular nature of the amino acid changes we saw were very consistent with the arms race model, not with the buffering model. So at this point, we're still thinking that the interaction between BAM and Wolbachia is in some sense a manipulation somehow of the functions of bag of marbles in the fly that in fact is driving this positive selection. But we haven't nailed that down as absolutely, it's just consistent with that pattern based on these long-term molecular evolution results. Again, one could ask about, so we've illustrated that bag of marbles, even though it's a critical gene, it's not critical in all species. So it kind of raises this question of flexibility. And we had found earlier that if we look at the YB gene, this gene actually, these are where our nulls are at this point. If we look at the YB gene, it's present across the whole genus, but it's completely absent in the pseudo-obscura lineage. But this is a critical gene in Drosophila melanogaster. So it's like, wait a second, here's another case of a gene. In this case, it's just simply gone. It's not that it doesn't function in that function, it's simply gone. And so we're now in the process of trying to take and look at a whole bunch of other species to see if this is an example of essentially kind of a genetic redundancy, whereas some people on developmental biologists side refer to this as developmental systems drift. We have the same phenotype being required, production of gametes, males and females across all these different species. We have to do that, but maybe we've got different combinations of genes that contribute that in different species. So what Luke Arntz is doing in the lab right now is he's taken candidate genes that we know by RNAi are critical to germline stem cell function, 366 of them, and basically taking and looking at those with regard to a variety of orthology predictions from ensemble compara, but also doing reciprocal best blasts to make sure, and look at synteny to make sure if the gene is predicted to be absent, is it really absent or is it not? And then testing these by PCR, so it's complicated. But in fact, the take-home message of this is, and this is an important thing to kind of worry about in some sense, is that if you take and look at these patterns, if you look very carefully using particular reciprocal best blast, you get a very different patterns from simply ensemble alone. But the take-home message is that some genes, actually like YB, are in fact truly absent in this lineage, and they're present in all these other species. If you look at BGCN, it's present in all the Drosophila species. It's absent in the housefly, but in the sheep blowfly it's present. If you look at bag of marbles, it seems to be unique actually to the genus Drosophila, and is absent from sheep blowfly and the housefly as well as other species. So there's clearly a lot of evolutionary flexibility. If in fact, if you take and look at this is again a caution. If you look simply at the ensemble predictions, for example, of orthology, you'd predict, as you looked at much more divergent species here, you'd see lots of these are predictions are there's a lot of orthologs are lost. Well, in fact, that's really not true. If you look at things with reciprocal best blast and synteny, we actually know that a lot of those still are really there. But just to kind of wind up, we do have, in fact, quite a few patterns of, in fact, genes being lost across the phylogeny, even of absolutely critical processes here. So again, we are seeing, and it's kind of like the observations of genetic redundancy in a different perspective, we are seeing the fact that there are a lot of flexibility with regard to how do you get a particular phenotype across these different species. And we're at the early stages of analyzing this, but there are some protein patterns that emerge. It turns out genes that are involved in lots of interactions and so forth actually tend to be more likely to be flexible in terms of their presence or absence. So germline stem cells are, in fact, functionally flexible. We think on average about 8%. And in fact, it turns out that it's really important to make sure you're looking at synteny as well as a variety of other characteristics to really know that gene is present or absent. And so far, the relationship is kind of complex between the actual functional categories in terms of making predictions. So we're not at a stage where we can kind of directly predict which genes might be kind of missing. But it does provide, I think, a future kind of open area for analysis, because there's so many high quality genomes being produced now that we can take and look at the processes of adaptation in one species and now start to look in other species and ask, have they adapted in the same way or different ways, or different genes? Is the complexities that Kristen and you guys have seen with regard to different, even populations of Drosophila melanogaster in terms of the genes involved in adaptation of particular things, is that complexity even amplified as we go to other species? Okay, so again, given the interest of time, I'm going to move very quickly. The take-home messages are that as your guys from talking to you today, I think you appreciate this. Finding the genetic basis of adaptation is hard. Linking it to what the environment sees is also hard. Even in something as simple as the bag of marbles gene, we thought. But it's important to do, obviously. In the case of bag of marbles, the genetics seems to matter. The ecology matters. Endosymbiotic Wolbachia is a part of it's a biotic environment here, life history, but also function. We're clearly seeing changing in function across the genus, and to the extent to which not positive selection is associated with that, we're still in the process of trying to tease apart. But nonetheless, there's lots of things to do. So the group, again, that's involved in this was at the SMB meetings in Ferrara just in July. My wife Wendy was at the meeting as well. Jackie, Catherine, Luke, Miwa, and myself. So this doesn't include the undergrads, but this is the core of the lab right now. So they're obviously the ones that did much of the work. So again, valuable contributions to them, and the funding from NSF or NIH, most of NIH over much of my career. And I thank you, and I'm happy to take questions. And I apologize for going a bit over.